Hello everyone, this is a PowerPoint about photosynthesis, which is part of the Bioenergetics section, Unit 4 of the Combined Science for AQA Trilogy course. So first thing I would like you to think about is what is the word equation for photosynthesis? So I'm just going to give you a couple of seconds to kind of think that. You might want to pause here and run through that equation for us. Hopefully you've remembered that the word equation for photosynthesis, so we have our reactants over here, which are of course carbon dioxide and water. Then we've got our arrow representing that a reaction has happened. And we have our products over here, which are glucose and oxygen. Now what we also have in photosynthesis is we have these things above and below the arrow. So we've got light energy or light above the arrow, and we've got chlorophyll below the arrow. Now they're there because they are not reactants. So they're not being changed in reaction in the same way that these two things are being reacted together to produce these two products. So we want to think a little bit more about these two substances. So chlorophyll is coloured green because it's a green substance which we find in chloroplasts, remember. And what chlorophyll does is it absorbs that light energy which falls on the leaf, and it means that the light energy is able to drive this process of photosynthesis. We can think a little bit more specifically about what light energy is able to do, because we know that photosynthesis is going to absorb this, so it's an endothermic reaction, and that light energy is going to be used to break bonds in our reactants, specifically in our water, and it is going to, that carbon dioxide and water are going to get rearranged, and they're going to end up producing our glucose as a consequence. The oxygen, as a product from the plant's point of view, most of the oxygen it produces it doesn't need. So what it does is it just releases it out through its stomata into the atmosphere, which is of course very useful for animals and all organisms that respire and don't produce their own oxygen, unlike plants. So that oxygen is just released out into the atmosphere where other organisms make use of it. So flipping this around, we want to think about what is the balanced symbol equation for photosynthesis. So pause video, you might want to note that down, or you might just want to say it out. The balanced symbol equation for photosynthesis looks like this. So we know that the formula for carbon dioxide is CO2. We know that the formula for water is H2O. We have to learn this formula. This is really important. So this is the formula for glucose. So C6H12O6, and then the formula for oxygen is O2. Balancing wise, well, let's take a step through this. So we have one carbon in a carbon dioxide molecule, CO2. We have 6 carbons in our glucose over here, so that means we need 6 CO2 to give us 6 carbons overall. With the hydrogen, we can see we've got 2 hydrogens per water. We need 12 hydrogens over here in the glucose, so therefore we need 6 waters, 6 times 2, which gives us 12 hydrogens. Oxygen wise, well, we have 6 times 2 here, so we've got 12 oxygen, plus 6 times 1 here, so 12 plus 6 gives us 18, and then over on this side we've got 6 over here, and then we get, so that leaves us with 12 more that we need. We've got 2 in an oxygen molecule, so 6 times by 2 gives us 12. 12 plus 6 is 18, so we can see that that equation is balanced. So moving on to a more general kind of idea of what photosynthesis is. So photosynthesis is an endothermic reaction. So that means that the products have more energy than reactants. And that makes sense because glucose is a store of energy. In actual fact, what we're doing when we're making glucose is we are storing that light energy that has been absorbed by the chlorophyll in the chloroplasts. And so photosynthesis is using energy, therefore we term it an endothermic reaction. That energy is absorbed as light energy from the environment, specifically from the sun, and it's the chlorophyll and the chloroplast that's doing that. So reminding ourselves of our structure of a plant cell, we can see that those green structures, green because they contain chlorophyll, are our chloroplasts. And underneath the microscope they actually look like this. So we can see a lot of chloroplasts in these plant cells here, and in the drawing above we can see them represented here. One of the things that we need to be able to talk about with photosynthesis is actually what the products of photosynthesis are used for. So we know that glucose is being made, but plants don't eat, so they need to obtain all of those same things that we do from that glucose that they're actually able to produce. The way that they do that is by converting or using that glucose to produce new molecules. Now, pictures are a really important way to help us think about what these things are. So we have represented here a number of images that give you the 5 uses of glucose that AQA expect you to be able to remember. So starting off in the top corner here, we've got this kind of lightning bolt that's going between these 2 electrodes. That would of course be energy. So the glucose that is produced by the plant can be directly respired by the plant in order to provide it with energy for all of its life processes. So for producing larger molecules or for reproduction or something like that. Moving down here, we've got representation of plant cell, and here we're particularly interested in what's around the outside. So yes, of course, that would be the cell wall, yes, and that cell wall, we need to know what molecule that's made out of, which is, of course, cellulose. So cellulose is essentially lots of glucoses joined together, and that gives us a molecule which you can build cell walls out of. Going down here, we can see we've got meat, egg, beans and peas, and so this is a really important food group for us, and plants need to be able to produce it themselves as well. So what we'll be talking about here is proteins. Now, proteins, remember, are made up of amino acids, and plants use glucose. They react it with the nitrate ions that they take up from the soil, and they produce amino acids, and then those amino acids get joined together by ribosomes in their cell in order to make proteins in the process of protein synthesis. Moving up here, we've got potatoes, and we've got this kind of floury stuff in the middle, which is another molecule that is made from joining lots of glucoses together. It's joined in a different way to the way it is in cellulose, and this is of course starch, because potatoes, beans, flour, pasta, all really good sources of starch, and that starch is insoluble first of all, so it doesn't dissolve in water, so it doesn't affect osmosis in or out of the cells. But on top of that, it's an energy storage molecule because it's just a way of storing lots of glucose for later times. So potatoes for getting through the winter, or other routes in which starch is stored as an energy source for later on in the year when there maybe isn't as much light energy. In the top image that we've got up here, we can see a whole range of different oils, and of course avocados are a great source of fats and oils as well, or to use their kind of more technical term, lipids. Now these things are a great energy store. In actual fact, if you had 10 grams of starch and you had 10 grams of a fat or an oil, say olive oil or sunflower oil or walnut oil, then we'd be talking about that fats and oils probably storing about twice the amount of energy compared to an equivalent weight of carbohydrates. So, what factors are going to affect the rate of photosynthesis? Have a quick think about that. So there are 4 factors that you need to be able to talk about, and again I've tried to attach some images to this to help it stick in our minds. We have carbon dioxide concentration, so remember this is the amount of carbon dioxide that there is within the atmosphere for these plants or within their environment. We've got temperature, so we know chemical reactions depend on temperature in order for them to react. We've got light intensity, so that's how bright the light is. So if we were talking about a bright summer's day, we'd say that light is very intense, whereas on a winter's day, we'd say that the light isn't very intense. And our final factor, this is one of the few places where this word is allowed, the amount of chlorophyll. So the more chlorophyll there is, then we can think about how that will affect the rate of photosynthesis. You need to be able to talk about how each of these factors actually affects the rate of photosynthesis, and particularly you need to think about how you could increase or decrease the rate of photosynthesis based on changing one of these factors. With carbon dioxide concentration, fairly obviously, more carbon dioxide means that there are more reactants are available for that reaction. So we need to think about chemistry here, and we need to think about that for a reaction to happen, we need the reactant particles to collide together. So in the case of photosynthesis, that would be the carbon dioxide and the water, and those molecules need to collide together, and they need to collide together with enough energy to react and form our glucose and oxygen. So therefore it stands to reason that if we have more carbon dioxide molecules, then the chances of collision between a carbon dioxide and a water are high. There will be more collisions, and so that means that we're going to get more reactants being turned into products, so the reaction is faster. We've got a little representation here, so some models and some images that we can be keeping in our head that help us think about this reaction. So here we can see we've got one carbon dioxide molecule, and it's reacting through this process of photosynthesis to produce glucose, and so just one molecule represents a low CO2 concentration. Whereas if I increase the carbon dioxide concentration, so I have 3 CO2 molecules, then in the same amount of time I can produce 3 glucose molecules as I did at the lower carbon dioxide concentration. So that model helps us to really visualize about why collisions are essential for reactions to happen. So temperature. Now here we want to think about collisions again. So increasing temperature means that particles are going to have more kinetic energy. If they've got more kinetic energy, that means they're going to be moving faster. And so we need to think about an analogy here. So if we would think about cars moving about, a demolition derby or something like that, and we had them moving about in an arena and we increased the speed at which they were moving, then it would mean 2 things. It would mean that the cars would collide more frequently, so there would be more collisions, and it also means that those collisions would actually be harder. So we summarize that in a reaction by talking about the fact that the particles are going to collide more frequently, and in particular those collisions are going to be harder. So we can use a particular phrase, which is that there are more successful collisions, and so therefore there is going to be a faster rate of photosynthesis in this case. So up here we can see that representation. So we'd have our carbon dioxide and our water molecules, and we can see that they need to collide together in order for that reaction to happen. Now one of the really key things with a biological reaction is that they are all catalyzed by enzymes, and we know that enzymes are very, very temperature sensitive. So if the temperature gets too high for the plant, then what that means is that those enzymes are going to get denatured, which means that the shape of the active site changes, and that means that the reactants, the substrate, aren't going to be able to fit into the active site as well, and therefore the rate of reaction is going to slow down and potentially even stop. Light intensity, remember we talked here about the brightness of the light. So if we have a brighter light, then that means that there is going to be higher light intensity, and so there is going to be more light energy available. That means that the chlorophyll can absorb more light energy, and so we're going to get more photosynthesis as a result. So we can see that down here with this model that I've got. So the green ovals here are representing our chlorophyll molecules, and we can see that at low light intensity we only get 2 arrows worth of light being absorbed, and so we're only going to get 2 arrows worth of glucose being produced. Whereas think about how I could model a high light intensity. Yep, that's right, more arrows, and so we can see instead of just 2 arrows being absorbed, here we have 3, 4, 5, 6 arrows of light being absorbed, and so we're going to get 6 arrows worth of glucose being produced, which is 3 times the amount that we had at the lower light intensity. Therefore, we've got a faster rate of photosynthesis because more energy is available for that reaction. So chlorophyll, so the only time in GCSE, or one of the few times in GCSE that you should use the word amount is in relation to chlorophyll. So if we've got more chlorophyll, then that simply means that more light energy is going to be absorbed. So let's go back to our model. Less chlorophyll, we've got 3 pieces of chlorophyll here, 3 ovals of chlorophyll, and that's able to absorb 3 arrows worth of light. Now it stands to reason. If I want to increase the number of chlorophyll molecules, that means I need more of my green ovals, and when I do that, that means that more light energy is going to be absorbed. So instead of 3, we've got 6, 10, so we have an over 3 times increase in the amount of light energy that is going to be absorbed, and so in the amount of glucose that's going to be produced. So more energy available means a faster rate of reaction as well. Right, now we can therefore begin to apply this and think about, well, if we've got photosynthesis occurring at a particular speed, then there are going to be factors that are going to affect the rate of photosynthesis. So only one of those 4 factors could, and if there's not enough of them, then that means that the rate of photosynthesis might not be as fast as it actually could be. It might not reach its maximum. Now, analogy for thinking about this would be something like a strongman pulling a truck, or you might want to think about if when you take your dog out for a walk, or your cat, or your rabbit, or whatever it is that you take out for a walk, and you're walking down the road, and you've got the lead on the dog, and the dog sees another dog, and what it wants to do is it wants to chase after it. So you're hanging on for dear life, and what that means is that you are acting as a limiting factor for that dog. You are preventing it from running off as fast as it can. You're like the harness and the chain that is around the strongman trying to pull this truck. So you're holding it back, you're stopping the reaction from reaching its maximum. And that's what a limiting factor is. So a limiting factor is the factor that is in the least or the lowest supply, and it prevents the rate of photosynthesis from reaching its maximum. So all of those factors that we've looked at are potential limiting factors of photosynthesis. Therefore, there can only ever be one limiting factor at any one point, and it's only by changing that factor are we able to change the rate of photosynthesis. Therefore, if we change a factor, so we increase the light intensity and the rate of photosynthesis increases, then we know that the light intensity was the limiting factor in that situation. Consequences of that are that if we think about those 4 factors previously, therefore they are all potential limiting factors of photosynthesis. So here they are again, those limiting factors: carbon dioxide concentration, temperature, light intensity, and amount of chlorophyll. So if you've got a piece of paper handy, it'd be good. If not, just think about what you would be expecting this to look like. So we've got sketch axes here, we've got light intensity on the x-axis, and we've got rate of photosynthesis on the y-axis. So what would you expect the graph to look like? Now, most people, when we do this activity, draw a straight line going up from this particular point. And that is unfortunately incorrect, or we could say that it's partially correct, because the line should look like this. So what we would see is that there is an increase in rate of photosynthesis in this region, and in this region there is not an increase in the rate of photosynthesis. So we're describing the graph. What that means is really there are two areas of interest. We've got the area before this blue line, where we can see that the rate of photosynthesis is affected by light intensity, and we've got this region where the rate of photosynthesis is not affected by light intensity. The line has leveled out, flattened out, or the technical term that we would want to use here is that it's plateaued. We can't say straight line because in this portion of the graph we can also see that there is a straight line. So a straight line doesn't indicate the fact that it's flat, that the gradient is zero. So we need to say that the graph levels out or it plateaus. Question for you then is, in this first portion of the graph we can identify the limiting factor, and we can say that light intensity is the limiting factor, but why do we know that? So we know that because when we change the light intensity, the rate of photosynthesis increases. Therefore, if we alter a factor and it affects the rate of photosynthesis, then we know that that factor was the limiting factor. Therefore, in the second portion of the graph, what is the limiting factor? What's the limiting factor in this section? Well, we can say that light intensity is no longer the limiting factor here. We know that definitely because we carry on increasing the light intensity, but the rate of photosynthesis does not increase. So something else is the limiting factor. This graph doesn't give us enough information for that, so we can't say what that limiting factor is with certainty, but it's one of the other four that we've looked at. So it could be amount of chlorophyll, it could be temperature, or it could be carbon dioxide concentration. The reason that we know this is, of course, the fact that when the light intensity increases, the rate of photosynthesis does not. Now you can do this for all of the different limiting factors, and it lends itself to some really good questions in your exams. So you can imagine describe this graph, explain this graph, and so you need to be able to talk about these things, and then you can potentially bring in some of that collision theory stuff that we've talked about previously. Where it gets a little bit more complicated is that kind of for higher tier students, they might think about talking about multiple limiting factors on a single graph. So here we've got a slightly different way of measuring the rate of photosynthesis. So we can see they're measuring it by the percentage of oxygen in the air. So the more oxygen there is, the faster the rate of photosynthesis, of course. So essentially this y-axis is a rate of photosynthesis. Down here on the x-axis we've got light intensity, and we're measuring that in arbitrary units. Now arbitrary units, it means it hasn't got any units, so we just arbitrarily assign them. So question wise, same thing as we had with the last graph. We have a part of the graph here, or two parts of the graph that we're interested in. We've got this part where we've got a positive gradient, so this first part of the graph, and for all three of these graphs it slopes off, it levels off a little bit earlier for the first few lines, but we have this portion all the way up here where we can definitely identify what the limiting factor is. And of course, if we're changing the light intensity and it is increasing the rate of photosynthesis, because we're increasing the light intensity, then therefore light intensity must be the limiting factor. So this graph's a little bit different because in the second portion of the graph we can actually, for the first two lines, identify what the limiting factor is. Certainly we know it's not light intensity because increasing the light intensity has no impact on the rate of photosynthesis, but we can identify it this time because the additional information we've got on the graph. So hint, look at these values on the different lines here. So for the 5 and the 20 degree lines, then we can say that the limiting factor with certainty is temperature, because when we increase the temperature we increase the rate of photosynthesis because we can see that the line plateaus at a higher level. What we cannot say is that for 35 it is temperature, and we can actually see that probably temperature is beginning to have less of an impact, and potentially if we go much above 35 we're going to get that denaturing. So maybe it's CO2 concentration here, or maybe it is the amount of chlorophyll that is affecting the rate of photosynthesis. We can investigate these rates of photosynthesis. So one thing that we might want to change here is the distance between the light, so this lamp, and the pondweed, and that's going to affect the light intensity. And so as we increase the distance between the plant and the pondweed, what's going to happen to the light intensity? It's going to decrease, and so we'll end up with a graph that looks a little bit like this. So as the distance increases, the light intensity decreases. So we can see we've got a graph that's got a negative gradient. And because of this, when we increase one factor, the other factor decreases, and we can say that these two factors are inversely proportional. So light intensity is inversely proportional to distance between the plant and the pondweed. So here we have how we can calculate the rate of photosynthesis. So it's the same as any other rate calculation that you've done. You can measure the rate of photosynthesis in different ways. So we say volume of oxygen or number of bubbles produced in a certain amount of time. But in order to calculate the rate from that, you need to know essentially the amount produced, so the number of bubbles or the volume of oxygen produced, and you divide that by the time taken. So we can see that represented within this set of data. So at 5 degrees C we can see we get 7 bubbles in 2 minutes. So therefore, in order to work out the rate, then we would want to do 7 divided by 2, because it's the number of bubbles produced in 2 minutes, which means that we would have 3.5 bubbles per minute, and that would be our rate. So have a go at doing that for the other values. So 15 divided by 2, therefore, of course, would be 7.5 bubbles per minute. 21 divided by 2 is 10.5 bubbles per minute. And for all of the others, 24 divided by 2 is, of course, 12 bubbles per minute. And so here we can see in the table that plateau. So in this region, the temperature was the limiting factor from 7 up to 20. But after 20, increasing the temperature has no increase on the rate of photosynthesis, so it's no longer the limiting factor. Now this all comes together into quite a complicated idea, and this is difficult. It's probably the most difficult bit within this entire unit of both photosynthesis and respiration, and this is the inverse square law. So the interesting thing is that when we increase the distance between the light and the plant, or in this case like a piece of paper, then the relationship we know is inversely proportional, but it's a bit more complicated than that. And I think it's a bit difficult to think about it directly as light, so I like to use a bit of an analogy, a spray can instead. So if I have a spray can here, and I press down the top of that spray can for 2 seconds, and I have a piece of paper that is 1 meter away, that cloud of spray paint is going to spread out over this square's worth of area, over this piece of paper, and so the colour on that piece of paper is going to be pretty intense. If I double the distance, then what happens is that spray paint has a further distance to spread out, and so it doesn't cover double the area. What it actually does is it covers 4 times the area. So my single piece of paper up here would only get 1/4 of the paint that would have been on the paper 1 meter away. So that means it's going to be a quarter less intense in terms of the colour. And so if we go back to thinking about that as light, and light is going to be a quarter as intense. And so that has a very important relationship that we can see in the data. Now, in order to work that out, we can use this equation up here, 1 divided by d squared. Sometimes the 1 can be a different number, so you can do it out of 1,000 because that makes numbers a little bit more friendly. We're going to have a look at that in a second, but let's just work it out. So if we do 1 divided by the distance 1 squared, so 1 squared is 1, 1 divided by 1, therefore means that the light intensity has a brightness of 1 here. Whereas here, we need to do 1 divided by a distance of 2 squared, so 2 squared is 4, so 1 over 4 is of course a quarter, so we can immediately see that. So the brightness is 0.25, or a quarter of what it was at 1. So that we can see those values, and here is an opportunity for us to do some calculations then. So for each of these values, work out the light intensity. So for the first one, you would do 1 divided by 10 squared. Right, the values here therefore are 0.01, 0.004, 0.0025, which we can clearly see is a quarter of the value there. And look at these numbers, 28 divided by 4 is of course 7, so we can see that that value has gone down by a factor of 4. 30 gives us 0.11, which we can see is a quarter of that because that was 4 carrying on after that. And so if we go 16 to 4, doubling the distance, then we can see that it's gone down by a factor of 4 again. And then our final value, 0.000625, which, as we can see, are less than friendly numbers. So sometimes you can see that this 1 can be changed to a different number to make numbers a little bit easier to work with. So all of this comes together in the idea of if you wanted to grow plants. So say you were a farmer and you were looking at producing food and selling it, then you would make use of this knowledge of limiting factors in order to make your plants and so your crop grow as fast as it could, so that you end up with as high yield as possible. But you have to balance that against the costs that go along with it. Because we know that in order to create maximum rate of photosynthesis for growth, we'd want loads of light, so you could install lights and have them on day and night to increase day length, and you could install them to make the light brighter so that you can so the plants are going to grow fast because they can absorb more light energy. But of course, to run those lights there is a cost because you have to pay for the electricity. You could also heat the greenhouse and you could add more carbon dioxide, but all of those things are going to cost you money, and so you have to balance the cost that is against the profit that you're going to make from your crop. And so there'll be a point at where you won't go beyond, because if you do, you will end up growing your crop at a loss. That's the end of the photosynthesis section then. So thinking about what you need to be able to do, you need to know those word and symbol equations for photosynthesis. You need to understand what the uses of glucose are in those 5 different ways. You must be able to talk about what a limiting factor is and how a limiting factor will affect the rate of photosynthesis. And you want to think about that required practical, understand it and know about the controls and how you process the data and how you could improve that practical. And as I say, the free science lessons video is really good for that. And then the final bit that you want to think about is how you can apply all of that knowledge into a situation where you would be growing crops, so greenhouse economics. Right, thanks a lot for listening. I hope that's useful.